DNA can be obtained from a variety of samples, such as hair, feathers, eggshells, saliva, bone, scats, etc. However, such samples usually have low amounts of DNA, which may also be degraded by environmental factors such as UV light, water, and decomposition. Adequate collection and storage of such biological samples increases the chances of obtaining enough amounts of DNA for genetic analysis. This instructional video illustrates how a variety of samples can be collected whilst in the field. At EcoGene, we've put together a DNA collection kit which we'll be using throughout this video. All the contents of the kit are commercially available, and you're encouraged to put together your own kit depending on your needs. The information contained in this video is used as a guideline, and pilot studies on storage and extraction of DNA are recommended before anyone endeavours on collection of samples for genetic-based projects. Dry samples are all those that are unlikely to rapidly rot. These include hairs, feathers, eggshells, plant material, and processed animal or vegetable products. The vast majority of these types of samples can be stored dry in a paper envelope or Ziploc bag containing silica beads. If the sample is a hair or feather, always manipulate them away from the follicle or quill, as DNA is obtained from these areas, and it's important to avoid excess handling, thus reducing cross-contamination risks. If you find more than one hair or feather, always treat them as different samples. It's important not to make assumptions about the number of individuals involved in any given case. When collecting eggshells, handle them very carefully to avoid any further breakage of the shell. If available, pick them up with clean forceps. In predator detection cases, DNA is obtained from the bitten parts of the shell. Laboratory technicians need to be able to identify such areas to carry out accurate swabbing. Processed animal or plant articles can be posted to the laboratory in their original packaging. If you're dealing with different items, always keep them separate. Items not protected by any packaging need to be placed in individual bags or envelopes. Fresh samples are all those that will decompose or rot. This mainly comprises tissue from either dead or living animals. These samples should be frozen or kept in preservation solution to avoid further decomposition and therefore degradation of DNA. Depending on the situation, you may want to send us the whole individual or a tissue subsample. If you are sending a subsample, cut a piece of tissue with a clean blade. If a scalpel is not available, clean the cutting tool of your choice with a solution of 10% bleach. Place approximately 1 square centimetre of tissue on the preservation buffer, for example, ethanol or Longmire buffer. It's important to label your samples with a unique ID in clear writing. An area where a carcass is found should be treated as a crime scene. It's reasonable to assume that whoever committed the crime left some evidence of its presence on the site. So examine the area and the carcass for any evidence of fur, scats, or any other biological material. For example, in cases where a bird has been killed by a mammal, examine the bird's claws and beak for any material that may have come from the predator. Collect such material following the recommendations from this video. We expect traces of the criminal saliva on and around attack wounds, so a swabbing technique is suitable to collect such saliva traces. Firstly, identify wound sites. Hydrate your swab with either sterile water or a preservation solution of your choice. It's very important that the preservation buffer or sterile water come in contact with only clean, new swabs. Proceed to swab the wound area, collecting more than 1 swab per wound, at least one from around the wound and another from inside the wound. Use individual swabs per wound. Allow swabs to dry and place them in their protective case and keep them at room temperature or, if available, inside a fridge. Remember to label your samples clearly. Scats are most often stored dried in preservation buffer or frozen. DNA from scats can also be obtained employing the swabbing technique. DNA recovery rates vary among species. Therefore, a preliminary trial to determine the best storage and DNA extraction methodologies is highly recommended before using Scats as a DNA source. Pick up Scats with clean forceps and place them in a vial containing a preservation solution such as ethanol, DMSO, and in some instances lysis buffer. Once in preservation solution, samples can be kept at room temperature away from light. Ethanol is widely used when air travel is not involved. If samples do have to be transported by air, lysis buffer may be a better option over DMSO, as it doesn't have the increased health and safety risks of DMSO. Scats can be kept dried in Ziploc bags with silica beads. Simply place the Ziploc bag inside out and use it as a glove to collect the scat. Then add silica beads to the bag. Silica will absorb the moisture, thus avoiding DNA degradation. For both the above techniques, the recommended amount of preservation liquid or silica is between 5 to 10 times the sample volume. Scats can also be sampled by swabbing. Hydrate the swab with either sterile water or a preservation solution of your choice. It's very important that the preservation buffer or sterile water come in contact with only clean new swabs. If a scat is fresh, hydration of the swab may not be necessary. Proceed to swab the outside of the scat. Allow the swab to dry and place it in its protective case and keep it at room temperature, or if available, inside a fridge. When collecting scats, it's important to know or at least infer information of the biology of the animal. For example, goats or rabbits excrete pellets, so that if a clump of pellets is found, it could be assumed that they came from the same individual, and therefore be stored together. If in doubt, always collect scats separately. It's important to remember that a pilot study is highly recommended before any genetic-based project is carried out. Such pilot study should compare different storage and extraction methods for material from your species of interest to ensure that enough DNA will be recovered to amplify successfully.